I want to introduce Dr. Randy Sweis. He did his undergrad and medical school at the University of Chicago, did his internship and residency in medicine at the University of Michigan, and then came back to the University of Chicago for his fellowship in hematology oncology, and he's now on faculty. His clinical expertise is genitourinary malignancies, so bladder, kidney, prostate, and testicular cancers, but his research expertise, which we'll talk about, is kind of drug and systemic therapy development. So I'll turn it over to Dr. Sweis.
Great. So welcome, everybody. Thanks for doing this. It's really exciting to give this kind of a lecture to people that are really interested in oncology. I teach the spring M4 course, Advanced Clinical Pharmacology and Therapeutics, but that's a broader coverage of different drug development in different disease areas, and very few to know people in that class are usually going into oncology or any oncology-related field. So my oncology biases are maybe not appreciated as much as they might be in this group, but so it's good to give this talk. This is actually a lecture that's essentially adapted from one of the talks in that class, and this is all M1s, is that correct? I think everybody's on mute, but anyway, I think this is all M1 students. So this is a kind of an intro, and what I want to do is give a little bit of a practical kind of, I guess, a practical view of the drug development process. I am going to speak to oncology a lot because that's what I do, but also have experience in general in drug development. Prior to medical school, I worked at Merck actually in medicinal chemistry, and I was not working on oncology targets at that time. I was into metabolic diseases and like diabetes and obesity, and we were actually at the earliest stage of synthesis of molecules that would go on to testing, and we made a few that ended up going into phase 1. Nothing ever panned out long term, but that was a really eye-opening experience and fun to be part of that. Then I went on to my clinical training and sort of with that background have now sort of interfaced as faculty at the interface between the lab and the clinic. I do phase 1 trials. I run a basic science lab as well, looking at mostly oncogenic drivers of immunotherapy resistance in the context of bladder cancer and sometimes skin cancers. And most of my clinical research is in early phase therapeutics, either phase 1 trials or later phase trials focused on GU oncology. So let me just jump in. So and I'm not sure, I don't know, people have different understandings of drug development. So if something is oversimplified, forgive me, but I think it's good to get everybody on the same page. This is the general process, and most of us have heard of phase 1, 2, and 3 trials and how that works, but I'll get into a little bit more nuance. So first of all, if you start off on the left here, prior to ever going into humans, there's a whole lot of preclinical research that goes into developing a drug, and that's a whole course in and of itself. But I'll summarize some of my experiences. So first of all, you have to have targets that you're attempting to engage. So the identification of new targets starts really with true basic science research, and I always emphasize this as the starting point. There is no shortcut. Everybody wants to develop drugs quickly. This is even more evident now with COVID-19 happening, and we want to do things tomorrow or yesterday, really. And I understand the urgency, and there are ways we can sort of push things forward quickly, but ultimately it requires a lot of basic science. And in oncology, the big revolutionary thing that's happened in the last 5 to 10 years has been the development of checkpoint inhibitors, in particular PD-1, PD-L1 inhibitors. Now, if you study the history of that, that was, that seemed to be such a rapid sea change that happened. We have approvals in 15 different cancer types now. But CTLA-4, which is the first checkpoint that was identified as a negative regulator of immune response, actually a lot of work came out of U Chicago identifying that. But if you go back to the basic literature, this protein was identified in 1987, and the negative regulation, the identifying it as a negative regulator, that came in the 90s. So we're talking about 20 years of basic science research before we ever could develop an antibody that can target it. In fact, therapeutic antibodies were not even out, until, in widespread use until trastuzumab for breast cancer kind of came along, and that was a big one. And then Avastin, targeting VEGF, that's the bevacizumab. So even the development of the approach to target these things didn't exist at the time these were developed. So a lot of things have to come together. I emphasize that just because that left side box is really big, and a lot of it is shots on goal. We have to do a lot of research in a broad array of basic science to really develop new targets and understand what would be a good strategy to try to therapeutically alter the course of a disease or prevent the disease. So, let's say you have the target now, and then you develop a drug. Then what do you do? So first of all, you have to demonstrate that the drug is hitting the target in preclinical models. Typically, this is like small mammals, like mice. And if it starts to look promising, actually before that, you're sort of doing target engagement studies in the Petri dish, in well plates, in a lab, in vitro lab testing format. And then into small mammals. And then you're really assessing early on, even in the animal models, for toxicity, potential for toxicity. You run screens to make sure you're not hitting off-target activities. And then, that could potentially cause side effects. And then you're even doing assays in animals for potential side effects. As an example, there was a model for emesis. So I was developing a drug for diabetes and obesity, and the target was cholecystokinin. It's CCK1 receptor, and one of the known off-target activity of the targeting that receptor was nausea and vomiting. And so we had a model with a ferret that would actually, they throw up very easily apparently. So you'd give them and count the number of times they threw up, and you test multiple. If you had a few different compounds that looked interesting, you'd test them all in that assay, and which one caused the least emesis, you would move forward to the next step. So along the way, you're assessing toxicities. This is all before you ever get into a human, and then all the way up to primates, where you're doing pharmacokinetic studies, and also you need to not only develop a drug that hits a target, but it has the appropriate half-life and such, and doesn't hit the other targets. So these are things that are done in the preclinical stage. And once you have all of that together, and you're really confident this drug is worthwhile to spend, $50 or $100 million on a phase 1 study, you might then proceed with an IND. So this is showing some of the technical documentation and things that are needed in the development process. And an IND is called, it stands for Investigational New Drug. And this is something that, so I'll get into that in a second. Let me stop here. This is maybe any questions so far? I've tried to summarize, how you do 15 to 20 years of research prior to IND in a few sentences. But I actually, I'm not sure if I can, if someone does have a question, just speak up, because I don't know if somebody's like raising their hand or whatever. It's hard for me to see on my screen.
Yeah, Randy, I'll let you know too if I see a question come across.
Okay, sounds good. So in an IND now, these are when you submit all of the your projected clinical trial plan to the FDA, and this is where they have 30 days to basically respond with comments, and they may and if they have no issues, they'll send you a letter saying okay to proceed. I have a couple of these for trials that we have open in phase 1 right now that were developed by me or colleagues and myself in-house. So these are what we call investigator-initiated trials. And when those are, when you develop a trial yourself, if you're an investigator in the future, you would synthesize the data on the drug and submit it as an IND, and then you'd get to hold the IND under your name and institution. Most of the industry-sponsored drugs, so if the drug companies develop a drug and you're running the trial or you're the PI at your site, they will hold the IND. So the industry sponsor will submit the IND and manage that. One thing about INDs that's maybe not clear to people is, obviously if you have a brand new drug, you submit an IND, but also if you have an old drug and you're using it for a different purpose. So this goes along with the label. So if you have a drug that treats breast cancer, you theoretically, I can't prescribe it for I could prescribe it for kidney cancer, but if it's not on label, a lot of times the insurance companies will deny it, and somebody might question why I would want to give it anyway, even if there is some preliminary data out there. If you really want to get the indication, you have to develop the study to show in that indication what your, not just the safety, but the effectiveness in that particular indication. And so what happens is, for drugs that are even already on the market, for instance, you might submit another IND that's for the use of that drug in a different context or in a different combination. For instance, here, and Dr. Golden's very familiar with the combination of immune therapy, pembrolizumab plus radiation. That was an investigator-initiated trial by a colleague of mine here, and in MedOnc and RedOnc together, that developed these protocols, and they had to submit an IND and get approved. Pembrolizumab has been on the market for 6 years now, but you still have to submit an IND because it's not been formally tested in conjunction with radiotherapy. So that was done, and IND was held here, and that study was actually published a couple years ago in Journal of Clinical Oncology. So IND is for new drugs, for existing drugs with in different combinations, or in existing drugs for different indications. Make sense? All right. So in phase 1, this is the as I think most people understand this next part, but phase 1 is typically safety. It's changed a little bit with at least in oncology, in terms of phase 1s have become very large now and often include expansion cohorts. They're kind of phase 1/phase 2 together, but they still all initially start with a safety period where you're testing a drug at a dose, and in going kind of slowly, usually giving the drug to about 3 patients at a time, and then spacing them out. And if they don't develop any major toxicities over the first 30 days, for instance, then you can proceed with the next 3 patients, and you often will escalate the dose to understand, what is the highest tolerated dose that you have, or what is the biologically effective dose. A lot of these studies now include biopsies of tumors in oncology that look for on-target engagement. It's unclear how much that actually drives dosing. We actually wrote a paper on that a few years ago, but for the most part, you're looking both for safety and target engagement, pharmacodynamic effects, and pharmacokinetic effects. So pharmacodynamic effects are basically what the drug does to the body, and pharmacokinetic effects are what the body does to the drug. So how quickly it clears, how it's cleared, whether it's through hepatic or renal excretion. And then the PD, the pharmacodynamics, is what it's doing to the body. And in oncology, that's usually intratumoral biopsies, but also systemic treatments, especially in the era of immuno-oncology. We're measuring a lot of systemic effects as well, effects on different immune cell populations, effects on cytokines systemically and in the tumor. And, of course, we are, and then underlying all of that, we are looking for responses. We're hoping for responses, but that's not the primary endpoint of phase 1. And phase 1 patients are often patients that have had numerous therapies and are, although it's not always the case now, because, some of these are combination studies, and so we try to bring them on to phase 1 earlier. But nonetheless, it's a population that has very refractory cancers, and is not often easy to find good responses with these drugs. But the key is identifying a safe and tolerable dose, and maybe seeing some pharmacodynamic effects, so you can move on to study it a little more formally in the phase 2. And this is often just done in one big study as sort of an expansion. They call them expansion cohorts. So if there's a study that looks promising in lung cancer, then you might expand for another 30 patients in lung specifically. The early phase studies are usually all advanced solid tumors, meaning a patient with any refractory cancer can enroll. And then if you think that you've had a couple of responders, for instance, in lung cancer, you might expand that cohort. Or sometimes they pre-specify cohorts that biologically they believe will be, investigators will believe that it will be most effective in. So, and then you expand your cohort, and these are typically single-arm expansion cohorts. You really are looking for some signal. You can't determine, and this is driving me nuts a little bit with COVID, because how many single-arm studies are you seeing with COVID where, we treated 20 patients with XYZ, and 16 of them got better and went home. Well, I don't know what that tells you, because if you did treatment ABC, you might also send 16 people home. What's the comparison? How many are going home with no treatment? I don't know. So this is true with oncology. So it's not meant to be a definitive study for effectiveness, but you are looking for some signal of activity, some tumor responses, and response rate is a common endpoint for these early studies in oncology. It's, in my view, a terrible endpoint for a large phase study, or even, some of these studies are getting FDA approval because it's typically based on radiographic changes, but that doesn't necessarily correlate with extension of life or improvement in quality of life, which are really the key endpoints for cancer patients that they want to experience, either live better or live longer, right? That's the ultimate goal. But response rate is a decent enough surrogate to look at early on. It's not enough to tell us, yes, this drug is amazing. And sometimes you see that a little hype that happens with these early phase studies. But we are looking for typically tumor response rates by radiographic criteria in a phase 2, for instance. And sometimes you might even do a randomized phase 2. And occasionally, I can think of a recent instance in kidney cancer where a randomized phase 2 actually led to approval. And it was 2 drugs that were already on the market, but there was a small randomized phase 2 that looked at in frontline kidney cancer between cabozantinib and sunitinib, which was, they're both TKIs. They both work similarly in kidney cancer, and it looked like progression-free survival as another surrogate, but it was better with cabozantinib, and they got approval in the front line. Their previous approval for cabozantinib was in the second line or later. But that was kind of a phase 2 that pushed it up to frontline. But it's still questionable today which one is actually better. It's not clear if cabozantinib is actually better, but it's at least was enough data that convinced the FDA to give them the frontline approval. So then phase 3, these are large studies, randomized most often. And they are sometimes for oncology, they are, in the hundreds to sometimes even a couple thousand patients. In other diseases, like just for perspective, like in cardiology, you might have a randomized study with like 50,000 patients. So it seems like a lot, but for oncology, they're actually relatively small. But that's usually enough to get a signal that's pretty definitive. And, it's going to be compared against standard of care. So something that patients often misinterpret is that like a randomization often includes placebo, and that's not true, except if the standard of care is supportive care or observation, then it would include a placebo often, but not if there's an active drug that is approved and standard in that scenario. We would never withhold a therapy from a patient, but you do have to have an appropriate comparator. To me, it's very simple. If you want to design a phase 3, you just think about the target population and you think about what you would normally do. What do the guidelines say? And that's what you do in that phase. And if it involves no other infusion or pill, then you might need to include an infusion or pill that's a placebo if your intervention is an infusion or a pill. If it involves a standard therapy like an infusion of X and your new drug Z is you think is better, then you need to do it in a placebo-controlled, double-blinded fashion in which the patients are getting infusions and you don't know which one it is. And they may, I'm sorry, in that case, it would not be placebo-controlled. You'd control against the standard of care. But it should be blinded in such that you don't know which one they're getting. And, there's a lot of more complexity to it than that, but you always want an interventional arm to have as little possible of difference compared with the standard of care arm. And so it gets a little complicated. Like for instance, in adjuvant treatment, so in bladder cancer, we have an adjuvant study. Right now, if you get treated with chemotherapy and resection of your bladder for bladder cancer, the standard is just to do routine surveillance, CT scans every 3 to 6 months. So that's easy in both groups. If you have a drug that you think is going to be helpful to prevent recurrence in the adjuvant setting, you still need to scan both patients at the exact same interval. So that makes sense, right? You do the scans at every 3 months or 4 months, whatever you define it as, and both groups should be the same. You also want to make sure that, in some cases, you can include a placebo. If the infusion or they're getting is every 4 weeks, you need to include a placebo. So, and that way all patients are coming in for infusions. And you might ask why, and it's because if you think about it, if you have a patient that is getting treatment and you don't have a placebo control, you might only see that patient every 3 months with their scans, but the other patients you're seeing once a month. So you're getting bias. You're introducing bias, and you're seeing your patients that are on the experimental arm more frequently. You may identify symptoms earlier, or treat or be watching them a little more closely, and you might miss something with a patient that's on the less frequent schedule. So you need to make the schedules almost identical as you can, and that includes either placebo or a standard treatment if there is one. Maybe that's obvious to everyone, but I just want to point that out. I'll stop. Any questions so far? Okay, and the last thing, which is confused a lot of times with IND, but NDA is different. That's a new drug application. That is at the end of your trials. You have a drug. It's definitely, it looks like it's active, and it is ready for prime time. You're as a company, or the company would like to market this drug. It's really a marketing application with the FDA that says, here's the data that we have efficacy in this disease, and we want to market it with a label that says this to this patient population, and look at all the data we have to support that, and that's where the FDA will approve or disapprove. The truth is the FDA is going to be involved all the way from phase 1, so it's not a shocker. We don't see a lot of shockers with decisions at the last stage. You don't go through 10 years of clinical development and then are shocked by the result. Usually if you're in phase 2, you start to see an activity. The FDA is involved at different meetings with you to design, in terms of you run your study design by them for both phase 2. There's sort of a phase 2 meeting and then an end of phase 2 meeting. And if it's really looking promising, you sort of confirm, okay, these are the endpoints we're going to look at in our phase 3. If we meet them, is this sufficient for approval? And they will say, yeah or no, you need to tweak it. Maybe you should tweak this or that. Ultimately, the company can do whatever they want with their trial. But if they have not really vetted their endpoints and their design with the FDA, then they're going to be surprised at the end, and they're not going to do that. They're going to want to know that they're designing it in the right way. And I can go on and on for hours about the different influences on design and philosophical or political perspectives on what should or shouldn't be approved by the FDA, but that's a separate discussion. The bottom line is, in any given time period, you're going to want to be in touch with the FDA in terms of what you're designing, in terms of what the goal is for approval. All right, we'll move to the next slide here. So I guess this is summarizing what I just explained. So this is in each phase, these are the key characteristics preclinically, and a lot of them are basically the same thing that we're doing in patients, but this is before that. We're doing it in animals and in vitro. So we're looking at the characteristics of the drug. And this is actually not so straightforward. When I was in drug development, we were working on small molecules, which historically have been the majority of drugs, but most recently now antibodies have become the majority of drugs in oncology. And even beyond that now, it's things like living cells, biologic, synthetic biology, like CAR T cells, chimeric antigen receptor T cells, or antibody drug conjugates, bispecific antibodies, T cell engagers, a lot of complex biological therapies now that you really have to understand the characteristics of what you're dealing with, which will impact the pharmacodynamics and pharmacokinetics of the drug. And sometimes you see a lot of misunderstanding of this, even at the clinical level for drugs that are approved. And as an example, PD-1 inhibitors or checkpoint blockade, when you look at the original phase 1s for PD-1 inhibitors, some of them like nivolumab, the on-target engagement, so the pharmacokinetics of the drug suggests that the half-life is about 25 days. So think about that. That means it takes almost 4 months before the drug is truly cleared. And when you look at circulating CD3 cells from the phase 1 studies, they found on-target binding to the receptor, even after the drug was cleared from your serum, out to 6 months. And I think that's as far as they looked. So these drugs linger for a long time. And what's even more important is that even when they're completely gone, because they're biological modulators, if you trigger an immune response, it may not matter if the drug is there. And why that's relevant is sometimes I see people on the inpatient side, residents and folks who are presenting a patient and they say, oh, the patient was on anti-PD-1 therapy for 6 months, but is now off. So this is, not immune-related pneumonitis, for instance. Now that kind of thing, by the way, is especially important with COVID because it presents the same. I say, when did they stop their immune therapy? And they say 4 weeks ago. I said, they're still on immune therapy. That drug is still in their system. And even if a lot of it is gone, the pharmacodynamic effect is still there. They triggered a bunch of T cells. You can get pneumonitis, months after you stop. So you need to think about, you need to understand the drug characteristics and the PD before you interpret how these drugs are acting in the patient. This is very different than a tyrosine kinase inhibitor for kidney cancer that has a half-life of 20 hours. If they have a side effect that develops a week after stopping therapy, that's probably not the drug, because that drug is completely gone. And it's usually they're targeting like the VEGF receptor, and that does not have permanent effects once you remove the drug. The side effects you see are dynamic. All right, so this starts from the preclinical phase, and then I talked about this. This is the IND. I'll get into a little more of
Randy,
the.
sorry to interrupt. There's a couple of questions.
Yeah, please.
In the chat
I've been
box.
talking for a lot of time, so I like to stop and make sure.
Can you see the chat box, or do you want me to read them for you?
Let me see if I can figure this out.
I can read them for you.
Yeah, because I don't see it.
So Ellen asked, how do you decide which drugs will be in?
Oh, I see it. Sorry, I can read it. Yeah.
Oh, yeah, go ahead.
I got you. So how do you decide which drugs will be in combinations versus, whoops, no, it's okay, versus on their own? So preclinical, yeah, good question. That's more of a driven by the scenario. So if you have for instance, what's happening now is PD-1 checkpoint blockade is the backbone of cancer therapy for about 15 different solid tumors. So most often for a true new target, like I have a phase 1 open targeting a new checkpoint that's called ILDR2. It's a target that's we're looking at it. And so that trial, for instance, was designed as monotherapy to look at the new drug completely on its own for safety, for dosing. And then there's also a phase where it's built in to include the combination of an immune checkpoint inhibitor with PD-1, because we think that might be synergistic. So it includes both. And so a lot of trials now will include both monotherapy. Ultimately, for any new drug, you're going to need to look at it by itself first, because you really need to isolate any new side effects. The combo trials are usually two drugs that already are on the market. For instance, they might be used in a different cancer, or maybe they're in a phase 3, but they're not approved yet for that cancer, and you want to see about combining them. So it's usually for drugs that are out there for a while, and we understand the profile and side effects. You wouldn't go in with like 2 brand new drugs together, because you wouldn't be able to interpret what side effect is coming from what. So for true like first-in-man studies, they're always single drug given at a time. And once you establish some safety, you can then start combining them if you think that's helpful. Hope that answered the question. Let me know if not. I'm going to move on to the next question. How does the process change in regards to funding, planning trials for drugs meant to treat diseases with high morbidity but low prevalence? So this gets to, I guess, like maybe hinting at kind of the orphan drug status sort of thing. That I don't know how folks are familiar with that, but there are some mechanisms within the FDA to try to accelerate approval. And orphan drug status is one, and that's for diseases that have high morbidity and low prevalence but high morbidity. And so there's not financial incentives for companies to pursue them. So the orphan drug designation gives you some advantages in terms of patent protection and market share. And I don't know all the nuances there, but there are those. And then there's also, I wonder, let me check my slides for a second. Because I think I have a slide on, this here. So I'll just skip ahead because this answers a little bit also that question. There are different processes. So let me just start by saying the regular approval process can take more than a year, even after you submit the NDA, depending on what it is they're reviewing. Now, if you're in a disease state that is and this happens a lot in cancer, where patients are in dire straits and there's not, they don't have other options. Sorry, I keep clicking. One second. Okay, I'm trying to pull up the chat and advance the slides. And again, it's all from screen. So there are multiple designations that you can use to accelerate approval, so to speak. One of them is officially called accelerated approval. And as you can see on the screen, it's for drugs that have serious conditions with an unmet medical need. And it does accelerate literally what it says. It accelerates the timeline. They review it. They guarantee, they will two things. It accelerates the timeline, but it allows you to use surrogate endpoints, and we can talk extensively about that. So surrogate endpoints are a way to sort of look at, in oncology, as I said, the key things that matter to patients are feeling better or living longer, but we use things like response rate, and that happens a lot. Accelerated approval can happen with response rate endpoints, so that'll shorten the time. But that's actually not based on the prevalence, which the question was asked about for low prevalence. This is based on need. So even a disease like bladder cancer, which is, got accelerated approval for, pembrolizumab and atezolizumab got accelerated approval based on response rate alone. But it's not because the bladder cancer is so rare. It's not that rare. There's 20,000 deaths a year in the U.S. The issue is there was no drugs. Nothing worked for patients who they had cisplatin-based chemo, and once they had that, there's really nothing else for the last 30 years prior to 2016 when they got approved. So they got an accelerated approval just meeting an unmet medical need. Now, so for these, I don't want to spend too much time on the subtleties. There's also fast track, and that comes on early. That's sort of as you're still in development, they can kind of outline a path forward quickly. This is happening for COVID. Breakthrough therapy, some of these overlap too, so you can have, it's basically like accelerated approval is like part of the fast track process and whatnot. It's not like 4 separate things. Priority review is the NDA step. That's where they guarantee 6 months or less, which again, I said, even after NDA, a lot of times it would be 12 months or so. And then I think, breakthrough therapy, that's another designation. But all of these are different mechanisms, and then the orphan drug status is separate from all these, but all of these are actually ways in which we can accelerate drug development for the purposes of rare unmet needs. And by the way, a lot of these developed from, I don't know if you guys ever saw, what was that movie with Matthew McConaughey about the AIDS in the 80s? Blanking, but anyway, the issue is like when HIV came along.
Band played on.
What is it?
And the band played on.
No, it was with all the illegal shops that were importing the AZT and stuff from Dallas Buyers Club.
Oh yeah, okay. Sorry.
So this was, the frustration. A lot of these pathways got developed in the 90s and 2000s based on the frustration of the FDA being so slow. And there's a whole, again, philosophical / political argument about, do you want the FDA to move quickly and approve everything with really crappy surrogate endpoints and put toxic drugs on the market that cost $200,000 a year for as many patients, and mandate that society covers the cost of that? Or do you do it in a more regulated way, but then have a slower process, or maybe more restrictive process that drugs that may not work for everybody, but potentially a small subset could benefit? That's the debate that we don't have time to get into. But anyway, these are mechanisms that were put into place to try to speed things up. So this was, let me see, where was I? So the next question was, how does funding, wait 1 second. Let me skip that because this, there's a follow-up to the last question about how do you recruit patients for completely new single drug trials if there are other potentially effective drugs available? Great question. In most diseases, healthy volunteers are actually doing the safety. Okay, got Dan's message. I will look at, and the band played on. That looks interesting. So in most drug development,
students.
Yeah, well, I'll take the advice as well. In most diseases, healthy volunteers are actually paid to test drugs at low doses until you get to safe doses. Oncology has followed a completely different path, and it's because historically these are patients that are in need, and we run out of therapies quickly, and patients are dying. So most of the phase 1 studies, in fact, as far as I'm aware, all of them are first-in-man studies are given to oncology patients directly in the diseases of interest. So it's actually pretty easy to recruit patients. And your question was whether if there's other potentially effective drugs available. So that's not when we're usually doing true first-in-man phase ones. We are it's patients who've exhausted all FDA-approved therapies. They've tried 3 or 4 drugs and nothing is working, and they're still healthy enough that they can participate in a trial. They have a cancer that's growing, but their functional status remains intact. They're still, they're still eating. They still have a reasonable quality of life, and they want to try something. That's the patient population that we are recruiting to phase 1 trials. It is a little different for the phase 1s that are in combination, because if you're giving it with a PD-1 inhibitor, you can then give it to a patient as a frontline therapy, really, if they have a PD-1 inhibitor that's approved, because they're not missing out on a sort of approved active drug. So we never withhold effective drugs that are out there from patients to do true first-in-man phase 1s, because the reality is no matter how smart we are with drug development, the response rate, the chances of having a cancer control with a phase 1 drug is single digits at best. There have been actually studies published on this. So effectively, you have a 90+% chance that this drug is going to do nothing for the patient. So you only do a true first-in-man phase 1 as a monotherapy if they've exhausted all known active therapies. Hope that clears it up.
Randy, I don't want to sidetrack you, but do you want to talk for a second about the idea of a therapeutic misconception in phase 1 clinic?
Yeah. There's what I just said about the response rate in phase 1 trials. That is it's very low, and as oncologists and patients and oncologists alike are often biased. We have a kind of an optimism bias where we sometimes the way we present phase 1s, it's not. It's actually been shown that it's often misinterpreted, and that a lot of patients that are on phase 1 trials believe that they are on them to get therapeutic benefit. And the true phase 1 objective as I said, we go back, it's safety. It's not efficacy. We're not actually even testing. I mean, we're keeping track of patients' responses and such, but that's not the primary endpoint. The primary endpoint of the study. So it's safety. And so the truth is that most of the drugs will fail. I don't know if I have the slide, but I do have a slide of, showing the, I don't know, but I can come back to this and just speak. As a preclinical, we develop 1,000 compounds. When I was in drug development, I myself self-synthesized 500 compounds targeting the pathway, and 2 of them got selected to go into phase 1 based on the dropout for other reasons. Then of the ones that go into phase 1, basically it's like of 1,000, 1 may get approved. So along the way, all of these drugs are going to drop off for failure or for toxicity. So the truth is, the therapeutic misconception is that in phase 1, patients will often perceive that they're on it for therapeutic purposes, and it's really a safety. So it's incumbent upon us as investigators to really do our best to explain why they're on it. And a lot of times, if you tell them, even there are patients that really just want to help drive the field forward. They understand it's a devastating disease and they have no other options. And even if they fully understand and they're cognizant that this drug is very unlikely to do anything for them, they actually want to participate and want to help the research agenda. And that's the ideal phase 1 patient. We don't want to be putting patients on that are confused about why they're on it or what the expected response rate is. There's several researchers I can direct you to some papers if you want. People have studied this therapeutic misconception and how it relates to phase 1 studies. Anything you want to add to that, Dan?
No, that was excellent.
It's a whole body of literature. It's hard to summarize.
Yeah, I mean, like Randy said, patients might seek out phase 1 trials all over the country or in the world and spend significant resources and time thinking they're going to get a therapy, but phase 1 is about safety.
Yeah, and as you mentioned, resources and time, that's actually a good point too, is that, patients are end of life. If a lot of them have less than a year, less than 6 months, these phase 1 trials are very involved. include a lot of visits, a lot of PK sampling, sometimes overnight admission to the hospital. So, time is a precious resource too. So that's often not accounted for. But we have to do our best to explain all of that, and it's all in the consent form. But as you may know, consent forms now are 40, 50 pages long sometimes. So it's how useful that is, I'm not sure.
Yeah, there's one other question that came in. What is the FDA doing for the year between NDA submission and approval?
Yeah, I'll come to that, but I skipped one before that on how does funding work for clinical trials. To what extent does industry, NIH, academic hospitals contribute to funding? And this is a great question. And I guess I would say the vast majority of funding is industry. Let's just say that up front. And the way the funding works is the studies are actually, if I open a study, if Bristol Myers Squibb comes to me and they have a really exciting new drug, they want to open a phase 1 or whatever it is, a phase 3, they'll come to us and and send me the protocol, and if we like it and we think it's going to help our portfolio and give options for patients and we think it's promising, we'll open it. And if it's designed by BMS and is a BMS drug and it's their they're the ones that developed it, then we will sometimes occasionally have some if it's usually in phase 1 or could actually be any of the phases, but if it's very early and they haven't actually opened the study yet, if you've been in the field for a while and they're asking opinions from leaders in the field, they might ask you for some advice on the design of the trial, but ultimately they're the ones writing the trial. So that's an industry sponsored trial. It's their trial. They give it to us. We open it here or not. We choose which ones we want to open. And if we open it, we contract with the company and they provide the drug and they provide payment for all of and every single activity that you do as part of the trial. If it involves extra EKGs or biopsies, they have to pay for that. The standard of care aspects of the treatment. So if it's routine blood work that we would normally get in the cancer condition anyway, like a CBC or a CMP, that's billed to insurance the way we normally would. But anything extraneous that's research-based is paid for by the company, and that's negotiated in a long contracting process, and that's how it works. So that's an industry-sponsored study. An investigator-initiated study, which is truly original academic research that's come out of I have an idea and I want to write this trial, and that could be I have a brand new drug that was developed maybe by a basic science lab at Chicago, and this is there's one in Red Ankh going through right now with this nanoparticle based that came out of labs here that are being looked at as a radiosensitizer. But that happens, and that is when you write your own trial based on the existing data. You have to have some a decent amount, the same expectations for preclinical safety and whatnot. But you design your trial, you write it, or it's an existing drug that's on the market, but you went to the company and you said, I want to write a trial and use your drug. Can you give me the drug? They might give you the drug for free. Or if it's approved and it can be covered and you're testing something else. You write your trial, you can get it through IND, but you also need somebody to pay for it, right? So that's where you can apply for funding through NIH. You can actually get even investigator-initiated studies funded from the pharmaceutical companies now. So that's a big source of payment. Now, they have to approve it and they have to like your idea. And the problem is sometimes they have to approve the trial. And so if they end up changing things in your trial and it becomes to the point where it's not addressing the questions or not of interest in terms of what you were interested in studying, then you have to decide, is it worth it? Do I seek funding elsewhere or do I modify the trial? So sometimes it's so you can get money from industry or you can get sort of independent grant funding. But we all know about grant funding has become very challenging, and but that is a source. I mean, there are and then there are other mechanisms that are non-industry supported, like going through cooperative groups that are supported by NIH, like Alliance, for instance, or SWAG or ECOG. These are groups that are that have some support for trials, and you can float ideas to the committees, and these are experts in the field that review these and prioritize trials. And if you get it approved, you can get it the infrastructure support that way. So there's a lot of ways that trials can be funded with industry, without industry, but most of the big trials you're reading are all industry sponsored. The phase 3s for sure, because these are, hundreds of millions of dollars typically. But a lot of you might have a phase 1 or 2 that's industry, that's funded through independent mechanisms. Okay, what is the FDA doing for a year between NDA and IND? NDA and approval. Sorry, why does it take so long? It's not just a queue. It's not just waiting in line. Let me see if I have a slide on. So this is, these are all of the different aspects that are involved in general. And well, okay. So this kind of, I had another slide that's not included, but how the FDA, let me see. Oh, here it is. Yeah. So this is actually, this is not exactly, but anyway, there are numerous aspects of things that need to be reviewed. And my colleague, I have, colleagues at the FDA that actually, one of them was a co-fellow here with me, and I've actually gotten some more of the inside, how things work. But, there are different groups. It's kind of like if you're on an internal medicine team in the hospital and you have a patient that has a lot of complex problems, you consult, nephrology, and you consult surgery, and you consult, and you get all their inputs, but ultimately you're the one writing the orders and you're the one making the decision. So in the FDA, there's different groups. And so there's like the breast cancer team, there's key central people that are involved in handling NDAs for, like, let's say breast cancer, but then they're going to receive feedback on all of the documentation from all of the different components. And one of them, might be clinical safety. So somebody just looking at toxicology, independent of everything else. One of them might be looking at, the manufacturing process and how they're going to actually make the drug and how they're going to, produce enough to sell. And is that, do they have that in place? The formulation of the actual pill or infusion. The other group might be looking at the efficacy data, and that would involve biostatistics and the quality assurance. So there's if you have trials with hundreds of patients on from dozens of sites, how do you ensure the quality of the data? And so there's ways to do that, which we won't get into, but somebody has to review that. So all of this to say is that there's a ton of work that goes into reviewing the application. And so that's why it takes so long. Some of it, and then some of it is because there are other higher, priority applications that come through these other mechanisms. There may be some queuing, but I just think it's actually a lot of work. I've for a small phase 2, I was writing up a study we participated in, and the final clinical summary report was 1,100 pages of PDF that summarized, and I think it was like only 40 patients or 50 patients on the study. So that included every piece of data on every patient that was in that study. Anyone that that even signed consent, even if they didn't receive a dose, they're included as part of the review process. So it's complex, but and anyway, so that's hopefully that answers the question. Oh, and by the way, just a little nuance to that. The PDUFA Act, which allowed user fees, that's very controversial. So in order for the FDA to expedite process, there was the PDUFA Act, which allowed there to be fees associated with submissions. And initially, it was like a small component of the funding for FDA, but now the estimates are it's more than half of the support for the FDA comes from PDUFA fees. So, which allows them to sort of move quicker because they can expand, hire people and keep things moving. But then, if you can imagine the conflicts that arise because they're reliant so heavily on industry fees, which are mostly industries that are paying that, then there's a potential conflict there. But that's a way that it's been sped up. How are the designations for new oncologic drugs made and are given understanding of the characterization of different cancers? Are more designations made based on genetic? Oh yeah, so I guess this is kind of getting to the question of the genomics of the tumor, and are we getting? So this is maybe a little area, and this is subject to opinion, but I'm of the opinion right now it's still a little bit more hype than in terms of developing precision oncology drugs that are really truly targeting a genetic mutation and not a breast cancer specifically or a prostate cancer. There are only two that I'm aware of, FDA approved drugs that are agnostic of tumor histology. And so I would say by and large, we are still developing drugs in our silos of diseases. Now with immune therapies, there have been, PD-1 inhibitors again are approved in over 15 different types of cancer. So that's broken down a bit, but each one of those approvals came from studies within each cancer type. And frankly, they are used in different scenarios within each cancer type. And oftentimes they're used in combination with drugs that are used in those specific cancer types. And the efficacy and toxicity is not the same in different cancer types. So I think they still do need to be studied in the specific cancer types. There are too many differences right now. Now, there are the two drugs that are agnostic or the two targets are the NTRK. It's one of these kinases that was identified, and it's very rare, but across any individual cancer. But there's actually a lot of patients out there that have these activating fusions or mutations in NTRK and TRK. And there's actually a couple drugs now for that. And the response rate was really high in patients that had the fusion independent across many tumor types. And so that was kind of the poster child for developing a drug that's targeting a true mutation. Now, I think, I would argue that that's a very unique circumstance. These are tumors that are truly driven by that fusion, and a lot of mutations we find are drivers, but maybe not the sole driver. And so targeting them, may lead to clonal expansion of clones without those driver mutations. But nonetheless, it's a great example of the precision oncology approach actually working. And if you do have a patient with this, regardless of your tumor type, you can use it. And I think the response rate was like 70%. Now, response rate, again, is a surrogate endpoint. So there's a whole other debate about, is it actually better than chemo? That's, it's not been tested, but nonetheless, these drugs are active and don't require histologic designation. The second one is PD-1 inhibitor for MSI tumors. So any tumor that is microsatellite unstable, not just colon, but you see MSI tumors of a lot of different histologies. And they have a very high response rate to immune therapy because, as you may understand from the biology, they develop a lot of neoantigens because they have mutations there's a problem with mismatch repair. So they generate tons of mutations, and they are more likely to be recognized by the immune system and subsequently respond to immune therapies that unleash the immune system. So if you have an MSI tumor, you can get pembrolizumab covered. And I think nivolumab, perhaps, there was another one that was approved as well, I think nivolumab. But anyway, so that's a tumor agnostic indication. Outside of those two examples, I'm not aware of any other so far. And most of the driver mutations remain unique to different cancer types. And we've learned over the years, for instance, with EGFR, that it works really well in lung cancer to target it, but not so well in other cancers. So I think it's a little of both. We need to have precision oncology, but it's also still, the histological subtypes remain truly unique, and the patient populations that carry those histological subtypes remain unique. I think I caught up on the questions, but these were great questions. So if you have more, keep them coming, because you kind of preempted most of my slides anyway. So I'm pretty much covered everything that was in here, which, this is just going back to the beginning. So here, the IND, we talked about this clinical development plan that will be submitted with an IND. An investigator brochure is a very long document and essentially includes everything we know about the drug. And it's updated after you go into the clinic with any new clinical information. And so it's a very long document, but it's really the sort of go-to gold standard to understand what drugs, if the patient develops a side effect, you can go to the IV and see, was this ever described before in any patient? And it will be in there. And it's updated regularly. The first in human protocol is what it says, very simple, straightforward, phase 1 protocol. The Yes, and maybe this is obvious, but you cannot start any clinical trial until you get FDA approval, and that's a big no-no. It actually allows for interstate transport of the drug too, usually, which is what is often needed, unless you're manufacturing it in your state. But that's the only way you can really move a drug across, and that's true for NDA as well. It allows for that, which otherwise would be a felony if you tried to move it. So this is the process I talked about. I'm not going to go through that. This is just an example of one of the INDs that I had submitted a couple years ago. Then this is what it looks like when I think this is cut off. Oh yeah, on the top here it says study may proceed. So this came 30 days within 30 days of my submission, and so then I was allowed to move forward. So when you have everything together, your protocol written, all that together, then you submit it, and then you prepare to begin your trial within 30 days. And they spelled my name wrong, but that's okay. We talked about, so in phase 1, again, key objectives: safety, dose finding, biological effects, and PK. Again, you don't see efficacy here. You track it, but it's not a primary endpoint. Again, normal volunteers in most diseases, except in cancer, we use special populations just because cancer is a terrible disease for many patients. Different mechanisms for approval, let's skip that. Phase 2, here's when you start to see therapeutic efficacy showing up. It's not a definitive test, but you look for it. You also might explore a couple doses. So even though phase 1 is the safety finding, you might find that your 2 highest doses were tolerable. Maybe you want to test, do you need to go that high, or there were you can do sometimes randomized phase 2 based on dosing. Or with the biologics, you have a question of intervals. Sometimes you do every 2-week dosing, every 3-week dosing. You might test that in a phase 2. And this is where, regardless of cancer or not cancer, this is where you're really in patients with indicated disease. So if it's a hypertension drug, you're starting to treat active patients with hypertension, not just normal volunteers, healthy volunteers. You have an end of phase 2 meeting with the FDA, and that's where you move forward with your pivotal trial. That's the phase 3. It's going to lead to approval or not in the proposed indication. Phase 3 is phase 3, pivotal trial. Safety, by the way, is always part of every trial. So it's not like you stop, you actually track every, and in fact, it's after you're approved, they have to monitor safety. And let me see if I don't have anything on that, but phase 4, if you ever have a patient with an adverse event, you can submit it on MedWatch, and you should, if you have something that's unexpected, that's not, outside of something unusual, you can submit that, and they will actually they have to submit. This is for drugs that are already approved. The FDA will review every quarter or every 6 months for approved drugs, safety updates. And so that includes anything that's been reported on the market, and also case reports. So I've published a few case reports, and I've gotten letters from the companies asking for more detail, that they want to know if there was any adverse events. If you go, so if anything that you report as a safety issue can be reported at any stage, even after it's approved. And it may change the label. You may end up with like a black box warning if something's identified later on. So, we approve drugs based on 1,000 patients being tested, and then you go and you start treating millions of patients. So there are things that develop, and it's really incumbent upon the clinician providers to report these things, and when they see them. And for investigators, you can even sometimes publish case series or look at these retrospectively in trial data if you identify some new signal that's worth noting.
As a quick side note, that's the reason it takes a while to do development of a vaccine, which is pertinent obviously right now with coronavirus, right? Is if you vaccinate 1,000 people, they're fine. If you vaccinate 100 million people, and you have a 0.001% risk of severe side effects, that's a problem. So
yep. Yeah, vaccines, they're even more widely used than cancer drugs if they become standard. So like coronavirus, if it worked, we'd want everyone to get it pretty much. So that's a lot of people. NDA, I talked about since 1938. The history of the FDA is very interesting, and I wish I had more time, but yeah, take my class fourth year. We have a lecture on that. It gets more into that, so how it was developed and the historical cases that led to a lot of this, where people were basically killing people with selling therapies for things, but prior to this. Anyway, this is all part of the NDA. Safety and efficacy, and then the labeling too. Again, so they have to approve the labeling. They also have to approve any marketing materials. You know, they have to make sure that the marketing materials are balanced. That's why you have that you know, really rapid speak at the end of the commercials for Opdivo, where it could cause death and could cause all these things. So you have to spend like the equal amount of time talking about the side effects as you do about the efficacy. So they assess all of that, yeah, and that's all the slides I have, but I can talk a little more about other things, and if in the remaining time, I don't know how much time we have, but the one other thing I'll point out is I get asked a lot in my class about herbals and supplements and where those stand, and the FDA doesn't regulate them in the same way as drugs marketed for a specific indication that are synthetic, so to speak, but they do regulate them in a way, and for instance, if there are reports that something is toxic, they can definitely remove it from the market. But they don't have, you know, they can also, you also can't market, you know, if you have echinacea or something, because it's derived from a natural source, they will allow you to sell it, but you can't say this is a cure for cancer, or this is even a treatment to mitigate cancer. They can only say certain vague language, like it promotes health, or, you know, promotes general immunity or something like along those lines. And so that's why you see that language on a lot of those. If they push too hard and start to be more specific, then they're marketing it for an indication and they don't have the label to do that, then, or the you know, FDA NDA that allows them to market for a specific disease, then they won't, then they'll ask them to pull it or change the label. So that's one way. And then of course, if there's safety signals, they can just pull it from the market altogether. But they don't regulate the dosing. They don't regulate even what's in it. And so, you know, you might see a bottle of ginkgo and it might have, you know, a variation of 1,000-fold from if you test every pill in there in terms of what's in it. Some of them may have almost nothing. I think that they are required to have some amount of the drug, but it doesn't have to be any specific amount. It could be like, you know, 100-fold or 1,000-fold less than what is thought to be a therapeutic level. So, so I don't have a slide on that, but that's an important aspect, and it's becoming increasingly used in our, you know, population. You know, you go through Costco, and 20% of Costco is supplements, so it's obviously a big thing.